Welcome everybody to the February edition of the DEBRA UK webinar. We are extremely lucky today to have Dr. Kevin Hamill from University of Liverpool, who's a senior lecturer in eye and vision, and he works on laminin proteins in wound repair and squamous cell carcinoma. Today he's going to be talking about zooming in on laminin 332, how advanced imaging helps us understand EB. Before I pass it on to Kevin, just a quick bit of housekeeping. The usual thing, please keep the cameras switched off. All your microphones will be on mute. The webinar will be recorded. Please don't make any private recordings or take any screenshots. As Kevin's speaking, please submit your questions and then we'll be able to answer them towards the end of the webinar. Again, please provide feedback to us on research@debra.org.uk. And on that point, Kevin, I'll hand it over to you.
Okay. There we go. That looks like it's right from my end. Is that okay?
That's perfect. We can see your slides.
Cool. Excellent. I just changed computer just instantly before this, as you know, Sagir. So I'm hoping everything still works. So my name's Kevin. I'm a cell molecular biologist at the University of Liverpool, and once-time geneticist. And when I said I was going to deliver this talk, or when I was preparing it, I kind of just get to the point of saying I want to show off some cool stuff. And so I want to show you cool stuff about my favorite protein. This is a protein called laminin 332. I'll tell you why it's called 332 in a little bit, but I just want to start with this video. And it's, I want to say it's like 15 years, so maybe a little bit older than that. But at the time, it was probably the cutting edge of what we could do with molecular tools. So the grayscale picture is a phase contrast image of a cell moving across a glass dish. So pretty clear, this bad boy back and forth in the angle, but that's only because we're looping. You can restart that. But the bit we're interested in here is the green stuff. So the green stuff is laminin 332, my favorite protein. And what we've done is we've tagged it with a green fluorescent protein so that we can image it in real time. And in this particular experiment, what we've actually done is we allowed cells to make laminin 332 for a while, then removed those cells and put a fresh cell on top. And what we found was those fresh cells absolutely follow the path that's been laid down by one of their friends beforehand. So that cell's moving back and forward on there. And so this protein, this laminin 332 protein, it's kind of like a road, or kind of like a railway line, or maybe a slug trail, depending on your viewpoint. And the cells use it to move on. But that doesn't mean it's relevant for EB, does it? Actually, it does. So let's talk about that. So this image here is a cross-section through human skin. So what's happened is we've taken a biopsy from a person, embedded that biopsy, it's kind of like an apple core, in a paraffin wax block, and then cut a really thin section through it. And then we've processed it with three different colors. So in the blue is a nuclear stain. So this is a DNA stain called DAPI. And what you can see is we've got layers and layers of circles. So these are the nuclei of lots of cells layered up this way. So this is the outside at the top, and this is the outer layers of your skin. So this is the epidermis. We're down below, you've got far fewer cells, a much lower density. So these cells down here are fibroblasts, and they make collagen, amongst other things, that give skin and other tissues its kind of structure. So this is like your barrier on the outside, and this is your structural stuff on the inside. The boundary point, this line in magenta, is my protein, is laminin 332. So it's an antibody against laminin 332. And I think just looking at this one picture, you can immediately tell it's at that junction between the epithelium and the dermis. The green protein is there really just for orientation. It's a protein called fibronectin, which is an extracellular matrix protein. So a protein that's found down below, but not found in the top. Beautiful, cool, love it. So how does this relate to EB? Right, so I'm just going to show you the same picture top left, but just below it is a hematoxylin and eosin version of the of the same sort of structure. So this is just a different way of showing it, where now we've got different shades of pink. The bright pink, the fuchsia, is the epithelial cells, and this is the boundary point. And the light pink is the sort of collagen-rich layer, and then the sort of purpler color are the nuclei again. So the same idea. It's not the same section, but same idea. And the thing that ties this to EB is basically, if this doesn't work, then separation occurs. And what I think this audience probably knows is epidermolysis bullosa can be split into 3 different subtypes. So we've got epidermolysis bullosa simplex, we've got junctional epidermolysis bullosa, and we've got dystrophic EB. So the top version, the skin separation happens at the cells at the very bottom. So they break because their internal skeleton, their cytoskeleton, isn't functional, isn't very strong. In contrast, junctional EB, the cells are absolutely fine, but it's the linkage between those basal epithelial cells and the stuff that's below it is broken. So the laminin is broken effectively. And then with dystrophic EB, the boundary points, the breakage point is just a little bit lower. So these white bits are blisters effectively, and you have a little bit of tissue left above before you get to the cells. So the separation is happening lower down. And the reason it splits in different areas is because different genes are mutated. So let's go back to this section, and now I've put a yellow box on it, and that yellow box is way too big compared to the image on the right, which I was kind of, we're zooming in a bit. So this on the right-hand side is a transmission electron micrograph image of the boundary point between the bottom of a cell, so this line here, and the stuff that's below the cell. And this structure here, this electron dense structure, is actually kind of like a spot weld that holds the skin down. So this is the structure that is non-functional in epidermolysis bullosa. I'll show you what's in it. But first of all, this is what happens if you took a section from somebody with EB, they put that hemidesmosome, this structure here wouldn't be formed. So a lot of work over the years studying genetic disease and studying protein function separately and together came together to identify the proteins that are involved. I don't need to remember these. It kind of sits like this, though there's probably about 1,000 different copies of each of these things. If I just break it down to the diagram, I just wanted to kind of layer on the EB phenotype. So EB simplex, you've got mutations on count 5 or 14, and that affects these internal parts. EB simplex often presenting with muscular dystrophy, though not always. You have mutations in PLEC1. And so this plectin gene, the plectin protein is involved in muscle function in addition to skin function. If you have mutations in alpha-6 beta-4 integrin, which is one of these across the membrane, this is the cell boundary here, by the way, you can have junctional EB, but it often presents with pyloric atresia. And down here, mutations in LAM-A3, so laminin alpha-3, LAM-B3, laminin beta-3, or laminin gamma-2, or mutations in collagen 17, you end up with separation here. And then with dystrophic EB, you end up mutations in collagen 7A. So this whole structure, the understanding how they bind to each other and like why this domain, it doesn't really matter for today, binds to here and why this binds to there, that's all been built up by protein chemists doing the interactions, while the connection with the disease state has been geneticists finding it. And together they formed this model over many, many years. Some of my supervisors identified the initial ones during my PhD, so 20 plus years ago. The same proteins are actually involved in an acquired disease, which you might have heard of, called bullous pemphigoid. So this protein is actually called bullous pemphigoid antigen 1E or bullous pemphigoid antigen 2. It's got multiple names. And that's and both of these are autoimmune diseases where antibodies that the person has raised against themselves that they shouldn't have done, like it's not, it's an acquired disorder, bind onto these proteins and stop it function. And they present with, bullous means blistering as well. So they end up with tight blisters or sometimes scarring blisters if it's down here. So a related disease, but yeah, I thought I was just mentioning on the way past. So that's the diagrams and the images. They all point to laminin 332 being required for that stable attachment, stable adhesion of epithelial cells onto the extracellular matrix, onto the boundary point. But you saw from the very movie at the beginning that it's not just about stable attachment. So this is an image of a mouse wound that is healed recently. So the damage was in the middle, and this area is new skin that's grown over the damaged area. So this bit over here is normal skin. So these are hair follicles coming down the side. Can you see you can see my pointer by the way. Oh, maybe I'll do that. Sorry, we'll make this more obvious. So that, yeah, these bits over here are hair follicles. And the purple again is our laminin 332. And what you can see here is in the area that's just finished healing, or maybe not quite fully healed over here, you've got lots more purple. So there's an increase in laminin 332 expression. And as we saw in the cells in a dish video, it's because cells move over this. They use it as a road to migrate to heal the wound. So if we go back to cells in the dish, this isn't a movie anymore. This is a static image where we've stained the cell skeleton, the actin cytoskeleton in red, and then you can see the snail trail. And what I want to kind of introduce is a concept that a protein that supports adhesion and a protein that supports migration, that actually makes sense, even though that they're ostensibly opposing functions. Because what we're really looking at is a sort of a carefully controlled differences in how tightly adhered the cells are. So if you have a strength of attachment that's here, you'll get maximal migration speed. You need to have some, but not too much, because if you have too much, you slow down and stop. So this is what you want in an intact tissue, but this is what you want in a moving tissue. And as Sageer said, this is also what ends up happening in a cancer tissue. In a squamous cell carcinoma, you end up shifting the dial toward the migration, and the cancer becomes invasive and metastasizes. Down here, no movement at all, because you need some attachment. So the cell needs to be able to bind onto something and then pull its way along. That's kind of like, if you put this in a sort of running analogy, over here it'd be trying to run in mud. This would be running on a race course, and this would be trying to run on top of water. You can't grip it, so you can't pull yourself forward. So this would be the Goldilocks zone, the ideal zone for migration, and there'll be another Goldilocks zone for adhesion. Okay, so back to the laminin again. So this video now it starts with a cell that isn't moving, and then it converts into a cell that's migrating over the dish. So we can see these kind of stepping stones where it was originally stably attached. And then over time, you'll see that it lays down some fresh laminin on the top. And the way we do these experiments, it's pretty cool, I think, to be fair. We take the genetic code for the gene, and then we take the genetic code for whatever color we want to look at. In this case, it was red, but I'm obviously showing it in black and white. And we basically clone the two together. And in this particular case, I used a virus to introduce it into cells so that they transiently express it. So the cell's chock full of it. You can see the cell is really dark, but you can see this pathway of it being laid down as it lays its road. But let's look at how cells move more generally, because there's actually for a long time, and I think it still exists, a bit of controversy about whether the laminin's coming out the front, as in being chucked out the front like you're laying the road down, or whether it's a snail trail being left behind and not actually used in the first place. So this is about, I want to say about 20 minutes of the movie when we're looking at a single skin cell. So this is on a glass dish, just moving. And I think I can watch this sort of stuff all day. I really love microscopy. So what you can see, the bright border here is the main cell body. So this is the sort of the most of the cells here. And then these extensions at the front, you can see sort of finger-like extensions, and then you can see this sheet-like extension. It's kind of like feeling its way forward. And so we can break down cell migration into multiple steps. So the first thing that happens is they have to transition from static to migratory. And to do that, they shift that adhesion dial down the way, make them less tightly adhered, both to the matrix, the stuff underneath, but also when there's a group of cells, a sheet of cells from the cells to each other. And then they change shape. So let's talk about front-rear polarity. So usually cells they're sort of top-bottom polarized. They have a very clear top that points the outside and the bottom that points towards the matrix. Now we need to go, right, we're going to go this way, so we're going to have some sort of sail shape toward that direction. And then we push out these sheets. So we call the thin ones filopodia, which just means finger-like, and then lamellipodia are the sheet-like extensions. So these two different structures you can see. And they are chock full of one protein called actin, and then they sort of grip onto the matrix and pull themselves forwards. So we call these pooling points, focal adhesions. And then by attaching the actin to the focal adhesion, the cell can then exert force, so traction forces to pull itself forward. And this is this image at the bottom left now. The red dots are one of the focal adhesions, one of the focal adhesion proteins, and the green cables here are the force-generating actin that's pulled against them. So you can imagine this being, the red dots being tightly adhered onto the substrate, in this case the laminin that's on the matrix, and then motor proteins pulling on those cables to pull itself forward. But it's all very well showing you a static image. Why would I do that? Oh no, this video's not working. Oh well, this was supposed to be a video that actually shows the dynamics of epithelial cells. Here we go. Maybe we can play it after. Yeah, so this is a very short movie. It's actually 15 minutes, but taken every 10 seconds. And what you'll see is the focal adhesions, the paxillin adhesions at the back dissociate and fresh ones form at the front. So we're getting new ones being laid down. And this is a cell actively, dynamically forming new adhesions and dissociating old ones. And it's all super cool. This over here, again, really complicated. It's there's hundreds of proteins involved here, but one of the proteins involved is laminin 332. So laminin 332 is bound by a different integrin, a whole bunch of chaperone proteins that then links it to the cell. If you take this movie and then just convert it into a temporal time map, it looks like a firework. I only put this in because I think it's a cool picture, where the early time points are blue here and the late time points are white, and it looks cool. I've actually got this on my wall in my office. Anyway, if you take an epithelial cell and stain it for the same protein for paxillin and laminin at the same time, you'll see that the patterns largely match up, which is cool. Okay, so this and a whole bunch of other data says that cells use laminin to pull itself forward and move. However, laminin 332 itself is also part of the off switch for this, for a different part of the wound repair process. So one of the features of epidermolysis bullosa is excessive production of granulation tissue. So not only do you have the blister, but it also fills with kind of gunk that sort of feels like a wound bed constantly. And part of the reason that happens is the signal to say, stop producing granulation tissue, stop producing this gunk is lost. And the reason I'm saying all that is actually some of the work that DEBRA funded right at the very beginning of my career, where I was actually doing sort of gene hunting, trying to find the mutations that cause these rare diseases. And for me, I started off my PhD journey working on this variant of junctional EB called laryngo-onychocutaneous syndrome. So these three words just actually speak to the different tissues that are affected. But the big thing here, so these are clinical images. We have nail beds filled with granulation tissue. We have the cornea, the front of the eye being filled with granulation tissue. You have throughout the oral cavity and then down the trachea, the larynx, it fills up with this granulation tissue. And then this is what granulation tissue is, by the way. And then the cutaneous erosions. Now, what you notice is they don't have the blisters. So this is, at first we thought, okay, it's affecting all the same tissues, but it's not the same as junctional EB. So DEBRA funded our research to investigate and find out what caused this and then why it happened. And that, to talk about that, and I just want to drop it in because I don't, this was quite a long time ago now. I don't talk about it very frequently, but so let me say, right? So the way we did it, we had 19 separate families who all had the same disease, and we gathered the DNA from basically everyone we could from those families. So the affected person, their parents, their brother and sisters, and anybody we could. And then what we looked for was regions of chromosomes that were shared with all the people who had the disease, but weren't present in all the people who didn't. And basically that involves thousands and thousands of PCR. Nowadays, it'd be super quick. You could just about do it just with a drop of blood from the affected person. You'd almost know the answer. In those days, it was an awful lot of pipetting that myself and members of the team did. Anyway, that took us to the laminin alpha 3 gene. And this is actually why I ended up working on laminin alpha 3 for the last 20 plus years of my career. So when we talk about genes, technically what happened was we had the location of the gene. There's actually some other genes nearby. But when we talk about genes, we don't think about them as a single box. They're actually made up of regions that make the protein and regions that don't. And we call the regions that make the protein exons, and the bits in between introns. So if I draw it again, we actually have 76, I haven't drawn them all. We have 76 different exons in this one protein. It's huge. Any biologist on the call will appreciate 76 is quite a lot. It's a big boy. But what's more interesting, or even more makes it, 76 is already a lot. It actually makes two very distinct splice isoforms. So that one gene makes two proteins that are quite different from each other. So we have a short form, what we call laminin alpha 3A or LAMA3A, and then we have a long form, LAMA3B. Now these two were known about from 1994, 1997, and I was doing this work roughly 2000. And so the human version of it had been annotated a little bit, but wasn't totally sure. So I was looking at the gene in like huge amounts of detail, more detail than perhaps anybody had done apart from like those early things. And what we discovered was that the mutation that leads to the strange granulation tissue disorder was located in one of these exons that is only found in the laminin alpha 3A form and isn't present in the alpha 3B form. So this is actually exon 39 of the gene. And when you compare that to all the junctionally B mutations, they all fall downstream of it. They all fall in this region, which you find in both proteins. And so this tells us almost instantly, or suggested almost instantly, that we've got different parts of the protein doing different things. So in the normal protein, it forms this trimeric structure with beta 3 and gamma 2, and that's fully functional, and that's fine. In the worst form, the more severe form of junctionally B, which used to be called Hurlitz, the protein isn't expressed, and the other two proteins don't get expressed. Whereas in this LOC syndrome, we're just missing the N terminus, and that tells us that N terminus is involved in regulating the granulation tissue response, which is pretty cool. Okay. However, I've kind of skipped a whole bunch of pain that was involved in actually finding this, because although the annotation is there now and the Genome Project is much more advanced, at that time it wasn't. It was incomplete, and we didn't find this mutation for an awful long time. I screened everything else and didn't find it, and then it was only later that we discovered this. And so because I was studying it in such detail, which you probably wouldn't need to do now, so I was a bit lucky in many respects that I noticed that at the other end of the protein, there was actually a couple of other proteins produced that no one had identified yet. So we identified these two proteins, and they both have unique exons that appear that was intronic sequence. And we went on to show that this one protein here that we now call laminin N-terminus alpha 3-1 is also expressed. So now we've got at least 4 proteins produced from the gene, and they're all different. So this short one is really good for supporting adhesion and controlling the granulation response. This long one's a big structural protein, and all the data so far says it's mostly structural. And then these new ones that no one knew about. So I went on and did some more studies on them. And what we found was that when you increase the expression level of this new protein in epithelial cells, they actually don't heal their wounds very well. They actually stop moving very quickly. So the left-hand side is a normal wound. We're watching cells move into that scrape. The right-hand side, they do move, but they struggle to get there, and it takes them a lot longer to heal that wound environment. Now, remember those hemidesmosome things that I showed you? Well, when we looked at mice that had extra LANT alpha 31, we actually saw that they had bigger hemidesmosomes. So they actually have much larger, probably, though these data don't show it directly, but they're likely much stronger attachment of the epithelial layers to the underlying matrix. So this protein seems to regulate hemidesmosome formation. And then when we look at that one live, so the left-hand side here is sort of like a cat paw. I think it's laminin beta 3 with the M-cherry tag, and you can see that it's really sort of the cells staying in one place. Where in the middle we've got the green protein, we've got the LANT alpha 3 one with the green tag on it. And what you can see is the distribution is quite similar, with these points on the edges forming up. And then this is the merge. So the green's the LANT protein and the purple's the laminin beta 3. And what we're seeing is these two proteins are interacting. A whole bunch of other data that's a bit more complicated show that they actually bind to one another. And the LAMP protein also changes the proteolytic processing of the laminin. So there's a whole bunch of extra stuff in here, but the overall picture is that this small protein seems to be converting laminin from a migration supporting molecule into adhesion supporting molecule. And so we've got this three different things controlled from the same protein with different parts of the same protein doing different things. It's complicated, and I appreciate that, but it's also amazing. It's like just you can see why this is well, you're beginning to see why it's so interesting. However, the real reason I wanted to give this talk is I wanted to talk about some of the sort of cool new tools that we've been generating to study laminin 332. But before I do that, I'm just going to show you a little model of how we think laminins assemble to form a basement membrane. And it plays reasonably quick if it plays at all. Let's try it. So what we have is laminins, which are these sort of propeller-shaped molecules that come in first to form a basement membrane, and they form these sort of hexagonal shapes where each laminin has got 3 different contact points. And then 2 other proteins come in, perlecan and nitrogen, and then they allow collagen 4 to bind over the top. I appreciate that plays quite quick, but I've got it in a static diagram here. And the reason that we draw it like this is the top right image. So the top right image is by Peter Yurchenko's group, and it was a long time ago. The left-hand side, this one here, is a sort of intact basement membrane that's been isolated. The middle one is what you get if you dissolve away all the collagen to leave just behind the laminin and some other proteoglycans, but mostly laminins. And you can kind of see that it's got this sort of hexagonal appearance to it. And the right-hand side is what happens if you dissolve away all the laminin and leave just behind the collagen. And then you see, okay, the sort of fibrillar kind of stuff. Okay, good. All right, why am I showing you all this? It's because this diagram here is the dogma. It's what everyone thinks. And it's supported by some really good data. When you isolate just these 3 fragments, they form this sort of triskelion motif. So when we draw it like this, which we had been drawing for hundreds of years, well, not hundreds, 15 years or something like that. And then recently, these data came out to show that they actually exist. Scale is important here, 10 nanometers. So that's, so a micrometer is 1/1,000,000th of a meter. 1/1000th of that's a nanometer. So this is 10 nanometers, so really small. But when you look at this, you're like, okay, that looks complicated enough. I actually look at it and say there's a problem. So the problem is that the laminin family is actually, they don't all look the same. We have many different laminins in higher organisms, and each have got different structure, and structure represents function. So the model works perfectly for the ones on the left, which look like this when you actually isolate them. And these are the laminins that you find in hair follicles in the stem cell niche, but not the major laminin in skin. Whereas the laminin in skin, the laminin 332 we've been talking about, is more rod shape, has only got a single arm. So as I think you can appreciate, it's pretty hard to make a triskelion with 3 different things binding to each other at a connection point if you've only got one. The data shows that this one can't really bind to itself, so it can't do it on its own. It needs friends to do it. So this has been a bit of an enigma in the skin and cornea and other epithelial fields for a while. And I sort of set about trying to address the question. And I have a hypothesis that I wanted to test. So lots of other people's work over the years have shown that the strongest cell surface binding sites are down here. So this is what we call the C-terminus, or it's just the bottom of the protein. Whereas the strongest binding sites for collagen 7 are up here. So this is very clearly, this binds the cell, this binds to the matrix. However, this red blob here actually contains binding sites that are a bit weaker for cell surface receptors. So technically this bit and that bit can bind the cell, whereas these 2 bits bind down the web. So this led to what is now my working hypothesis, that at low density, when there's not much laminin present, when you're making a new matrix, such as during the process of wound repair, the laminin binds to the cell surface with both ends of the protein at once. Whereas once there's a lot of laminin present, And when processing events happening, we get a change in orientation where the laminin protein points down the way and the major cell surface binding is all at this end, at the C terminus. Okay, mad hypothesis, why is that useful? Well, some of discovery science is about understanding the process. Once you understand the process, you can then mimic the process. And for example, you perhaps can unstall a chronic wound or perhaps replace the certain parts of the protein that are absolutely required for function. So that sometimes we do things because they're we don't know where it'll take us. But yeah, anyway, right. So this was my hypothesis, and I wanted to develop a way to test it. But at the same time, I'm develop better tools than have ever been available to study laminins with. And so the issue about testing it is a resolution issue. So that the whole protein from the top to bottom is about 70 nanometers long. And all the imaging I've shown you so far have used a pretty good microscope, a confocal microscope. And those microscopes have got a resolution of XY, so that's the horizontal plane. So we can tell two things are two things. That's what resolution means if they're at least 150 nanometers apart. Our bigger problem is the Z, so that's like the up-down direction. We're kind of limited to about 300 nanometers with even a very good microscope. And this is obviously smaller than that. So I can't tell if this or that is upright because I don't have the ability to differentiate from that height. And so we made some new tools. And what we've done is we've used the most advanced form. So green fluorescent protein and M-cherry, red fluorescent protein, are kind of old. They've been around for a long time now. The people who discovered them got the Nobel Prize, I want to say 20 years ago. And they're really cool. Like I still love them. But this is like the next generation. So this is a protein called MEOS 3.2. And what we've generated is one version of our laminin construct where we've tagged this end and another one where we've tagged the other end, the C terminus. A MEOS 3.2, and I'm sorry, this is maybe a little complicated for this audience, but is it was called a photoconvertible protein. So it starts off green, and then if we shine blue light on it, UV light on it, it changes and becomes red. And then if we shine more light on it, more UV light, it actually turns off. But then when we shine even more light on it, little individual proteins will turn on one at a time. So that is mad, and it's awesome and unbelievably useful. So this photo conversion from green to red can tell us that if you imagine a cell, right, we can convert stuff so it's all the protein is red. Then we know that any new protein is green and every old protein is red, and we can track them individually. So we can watch new deposition on top of old. So that's just useful on its own. But this ability to turn it off and then come back on again allows us to use a new form of microscopy called photoactivation localization microscopy. So this is a form of what's called super resolution microscopy. And what we do is we basically turn off almost all the protein, and then every time we take a picture, only 1, 2, 3 of the individual laminin molecules is on in that one picture. So then if we take a composite image of 30,000 images, we can get every individual protein on the dish turned on at different points. And therefore, if we're only imaging one at a time, suddenly resolution is not an issue anymore because you're only looking at one. So it can only be one. So it's actually, it's really cool. Now, we thought that was cool, but we thought we would go one step further as well. So we made a double tag version. And our double tag version, we used a little amino acid tag. So we put 15 amino acids. The whole protein is, I can't remember now, about 6,000 amino acids for the heterotrimer, about 2,000 for laminin beta 3. So this is quite small. And then we have a very, very, very small version of an antibody, a nanobody that can bind to that. So now we can localize the bottom in green or red, and we can localize the top using infrared, using far red. So what can we do? Well, I kind of suggested we could do dynamics, we can do turnover, and we can also do the super resolution. So just to show you another cool movie, because obviously I just love doing microscopy. I have great fun. So this is, you can actually see the stitching points. So we can zoom right in on this image, and this is a corneal wound initially getting ready to move, and then you can see a whole track. I mean, I don't know how well it's projecting on your screen, but you can see the cells laying down the road as they move into the wound environment. And the good thing about this compared to the previous one, it's stable. So you can see that every single cell is expressing. So this is actually allows us to do a lot of things that the old tools couldn't do. if I move on to these movies, which I hopefully will loop, the left-hand side, I just took a cell, dropped it on the dish, and then filmed it as it spread out. So when it cycles back, you'll see it starts off round. And so we're looking at the very, very first stages of laminin deposition of a cell. It's a bit grainy because it's actually quite hard to do this. This is the best movie we managed to get. And then the second one is if you left the cells on the dish for like 2 or 3 days and then started filming them. So you can see there's sort of a flower pattern in the center, and then cells come in from the bottom and begin to add to it and move around. And this is just using the green, by the way. This is not doing any conversion or anything. This is just filming it using the same sort of imaging modalities as we did previously. And we need to be aware of time here. I think I'm still okay for a bit. So you've got these things, you want to push them a bit further. And there's a couple of sort of relatively straightforward ways. There's sort of like different levels of super resolution microscopy you can do. So one's called ARIScan, which is really fast. And we can go from our standard sort of picture, which still looks beautiful. Like I love taking pictures of lamins. They all look very like rose-like patterns. But I hope you can appreciate that after we process it through ARIScan, we begin to lose some of the fuzz and we go in to get slightly better resolution. The gain's not huge. And obviously, the more you zoom in, the more useful it is in terms of seeing where things go. But the other one that I think is quite cool is called structured illumination microscopy. So again, our initial image isn't super well resolved, but then once we use what's called structured illumination, you can begin to gain resolution. And the way that this one works is we take about 12 images, but rotate a grid in between, or a lattice in between, and so this is a lattice sim, as it's known as, and basically every time you rotate it, which I think you can just about make out the grid in the unprocessed image, you remove some of the out of focus light, so you gain resolution. So this is actually looking at that N-terminal nanobody tag, and this is looking at the green, hence green. However, I wanted to show you that the structured illumination in a sort of larger format. So this again is the standard microscopy image, except now we're looking at the 3 different colors. So we're looking at this would have been green. So this is like all the protein. We've converted the top half of it to blue. It's red. The colors are all, it doesn't messed up, but this is when we converted it into the blue color, and then this is the nanobody. So remember, these two this plus that should equal that, really. And then we convert that to the structured illumination, and we begin to get better resolution, but we're still quite far zoomed out here. So this is a scale bar down the bottom right, and I've made it bigger here. So this is a 10 micrometer, 1/1,000,000th of a meter, showing that the 2 images layered on top of each other. And I'm going to zoom in on this square here to show you what we gain. So this was what, if we zoomed in on that area, would look like, or does look like, if you'd use conventional microscopy. But this is now using structured illumination. And what I hope you can appreciate is that we've got blue blobs, pink blobs, and then areas where they're connected by yellow blobs. So that is showing us that we can see the N terminus and the C terminus. We can see the top and the bottom of the protein being connected. And these distances all make sense in terms of the protein structure, which is pretty cool. Oh, by the way, none of this is published. This is all new. These tools are, anybody on the call who wants to use them, just give us a shout and we'll send you them. So cool. So Gary, have I got 5 more minutes that I could keep running for? Is that okay? So that I want to show you the coolest thing of all. So by the way, these are the microscopes that I've shown you images from before. So the live stuff, I've got these boxes on them so that we can study in real time. Top left's a confocal. This is a bit more sort of standard. This thing here costs about $250,000 probably, maybe 350,000. This was 250, and these two are about 100,000. So they're nice bits of kit, and these are all in the building that I work in Liverpool. But for the last few minutes, I'm just going to show you when we're pushing the limits to a whole different level. So I got really well, lucky, I guess, but also like I was super happy to get the chance to go and use the best microscope in the world for doing this type of things. And this is what it looks like, which when you turn up and you go, right, I'm going to use this amazing bit of kit, and you walk in the room and you go, oh, this is scary. And it is scary. Like I genuinely couldn't believe that they were going to let me use it, and also that I was going to be actually able to do it. So this is known as it's an interferomic photoactivation localization microscope. And you can see in the image on the left-hand side, there's just a few points of light. That's what I mean by each individual protein is only on at a certain time. We're looking at a handful there. The sample goes here. I don't know if that's even visible. I'll turn on my laser pointer again. Yeah, the sample goes in this little bit here, and then all the gubbins around here are lasers. The picture on the bottom left now is the Nobel Prize that the person who developed this microscope was awarded. So this is Eric Betzig, who works there. It's quite awe-inspiring having a Nobel Prize winner stood beside you while you're trying desperately not to break his machine. And this is me looking happy taking a photo of it. And this is it with the lasers on. Super cool. How complicated is it? Well, this is the diagram of how it works, and good luck. But basically, the sample goes in the middle, you shine light on it, and then you collect the light from two different directions at once, and then pass it through a beam splitter, and then that layers the light from each camera in very defined ratios on top of each other. And then we use interferometry to say that if a particle is closer to one objective than the other, then the pattern of the interference will be different. And don't worry if you didn't understand. I'm not even sure I do. Just to layer that together. Objective 1, objective 2. So there's objective 1 and there's objective 2, and the sample sits in there. This is also what the software looks like to drive it, which is scary. And then these are the various steps. So these are the actual interferomic paths of a variety of different spots. There are the spots changing over time, and these are the three different cameras, the wave pattern of an individual point. It's super complicated. And then this is a whole bunch of different steps of the analysis. A lot of this is still ongoing, by the way. So we've done the work, we've acquired the data, but the analysis is taking a lot. And that's what you get. So I think you look at that and go, that is not better than any of the other images I've shown you before. And you're right. It doesn't look all that cool at all. These bright spots are actually focusing beads. They're fiducial beads. So we know exactly where those are. So we use them as reference points. But the reason this is cool is when I do this. So again, it doesn't look much better. But now what I've done is we've color coded it based on height. So yellow is lower down than blue. And what you can see is around the edge of the cells, we've got blue, and to the inside of those patches of where there's the laminin, it's yellow. So that protein isn't flat. It's at a different height relative to the edge of the cell at different points. If you zoom in a bit, even across that area, you can see a bit better. You've got sort of like the turquoise over here and the yellow over there. And then down the bottom right here, this is the focusing bead, and then this is the range of the different laminin heights. It's complicated. I appreciate that. It's complicated for me. So I just wanted to show you that because no one's ever done this before, and it's and super cool. And so yeah, that's sort of where I wanted to end. The very last thing I wanted to talk about is a project that we're hopefully starting soon, because we actually can use some of these tools to help us design or find new drugs to treat junctional EB. So this is a more complex way of looking at the gene laminin beta 3. It actually runs in this direction. Those little arrows tell you the direction. And this mutation here is the most common mutation that leads to junctional EB. So this R63X is there's a hotspot. There's a region of DNA that's easily damaged, and so this happens more frequently. And what we're going to do is we're going to use what we've learned about this construct to introduce basically a fluorescent protein directly into the gene of the C-terminus. So everything up to now has been a virus. Now we know how to do it. We're going to do it this way. So we're going to introduce that in there. So the cells will be green, but then if we introduce this mutation, the protein doesn't get made anymore. So they won't be green anymore. And then what we can do is we can test the cells with any new treatments emerging, whether that's a drug or a genetic therapy, to identify whether it restores expression. So the way that would look, we could do tens, we could do hundreds at a time. So what we're going to do is 96 well plates or even bigger well plates, grow the cells in all of them, treat them all with the drugs at various concentrations. And then if we identify one that makes the cells go green, then we know it's turned expression back on again. So whether that's whatever therapy that is, we'll know that this whatever drug was in A6, we should then look at further and see if it is useful. So this is the future. Hopefully, if things go to plan, we'll start doing this in the next few years. But that's where I'd like to end it today. I showed you lots of pictures. Most of them were generated by people other than me. There's all the team. If you want to find out about our work, this is our lab website, and this should work. This should take you to the Lancet Laminin website. Our most recent paper came out last month describing the new switch. So that's the first thing on the blog, but there's lots of things on there if you look at the science room pages that are designed for sort of public consumption, so they're a little bit less complicated. And yeah, I think that's where I'll end my talk. So do you want me to stop sharing, Sagar? And
Oh, yes, please.
yeah. How was that? Too complicated?
No, that was great. It's a complicated subject, and especially with the fancy equipment you're using as well. I think things have moved on since last time I did any bit of confocal microscopy 15 years ago.
Yeah.
And well, there's the reason why I'm no longer in the lab. I was never very good, you know. So no, absolutely amazing pictures, and interesting to see that laminin 332 almost, well, not almost, lays the road down for the cells to travel along from point A to point B to close all the wounds.
It honestly, it's incredible. The question in the chat there, has this been done for other proteins? The answer is yes. Laminins are difficult to work with. They're big, which is the biggest problem for all these sorts of things. So a lot of the tools were developed to study a lot easier proteins to look at. So actually, there's loads of tools to study focal adhesions and their turnover. I've done some work using collagen 17 beta 4 integrin. So I should have put those in because they're the other hemidesmosome proteins, and but they're also pretty hard to work with. What I've kind of glossed over is the amount of pain that's involved. The number of times. So that first movie I showed, I went to the microscope so many times trying to get the perfect version of that. And in those days, the microscopes would go out of focus, and so basically I'd be sat there pretty much overnight, focusing, taking a picture, focusing, taking a picture. So I actually started with a relatively straightforward one to do, but the one that at various points I thought I was never going to get. So that's actually part of the reason why there is still controversy as well. So people don't necessarily know if it comes out the front or the back.
And I think another question has just come through.
They're asking for the transcript of the recording.
Yeah, transcript of the recording. No, we can potentially look at putting some of the recording onto the website so you can watch it later on.
Yeah.
No.
In terms of the transcript, we might not have recorded the transcript, I guess, but
I'll
probably isolate it from
yeah. I'll check the transcript, see if that's available.
Yeah, I appreciate I mumble and also say a lot of words that you won't be used to hearing, so that's a bad combination. I also like it's hard because I get into this stuff. It's yeah.
You can see you're absolutely enthusiastic about this, and you absolutely love it, and that comes across on your presentation.
And it's always been my favorite thing to do. So I talked about the LOC project, and I went down the sort of genetics PhD route because I thought I found genetics easy, right? Everybody finds something different easy, and genetics is complicated, but for whatever reason, it just clicked in my brain, and I really, really enjoyed doing that. And then everything I've done since then has all been like serendipity, following the science, which I'm sure you know what. I mean, it's what everyone's story in science is, that you do something, and so yeah, I did my genetics thing, and then at the back end of that, my PhD, and then the little postdoc that DEBRA funded, I got to do the first set of microscopy. So the movie of the lamp wound closure, actually no, that came after. I don't know. There's some of the I never like the temporal stuff of it. But anyway, yeah, whenever I stained my first cells with antibodies against laminins and saw the rose-like patterns that they were forming, I was like, I could do this all day. And then I ended up yeah.
I think a question come through. Is it possible to give a lay summary of the main headlines in terms of the progress treatment for EB?
Yeah, so there's a few different things in there. I spoke a bit about how we first identified the mutations that caused the thing before the different types of EB. So
Mm,
that work, without that, there would be no treatment at all ever
mm,
possible. So that's laid the foundations of every different type of therapy that's gone before. And that, most of that wasn't me. That was actually people before, but certain parts were. The new bits are we're going to understand the proteins in a way that no one else has ever been possible to.
mm.
Um, and it's sometimes hard to predict exactly how that translates to a disease, so to a therapy. Um, can I just share my screen again?
Yeah, yeah, sure,
Yeah.
sure.
Yeah, so um, this isn't about uh, EB, but I had this slide in the background. So by studying the laminins in the past, we identified that a part of the laminin
Ke-
improves.
Kevin, you're on presenter mode, so we can't actually see
Uh,
your slides.
what can you see at the moment?
We can see your slides, but they're quite small.
Oh, okay. Let me go to that. How about that? Is that better?
That's better.
Yeah. Okay. Yeah. So this is a rat where there's a burn wound being created in the back of the rat. And then what we've added on the bottom panel is a little fragment, a little protein fragment from laminin 511, which is part of the family. And what you can see is we have improved the rate at which the wounds heal. That's the graph on the right-hand side of the same thing. So basically, by 21 days, the control animals hadn't healed their wounds, but we made it faster by adding this fragment of the protein. Now, these are healthy. This isn't EB related. But we couldn't have done this experiment without knowing that this little part of the protein, which we've called E8, is involved in wound repair. So it's sort of like you understand the process better, then you can mimic the function to make things advance. So I appreciate that wasn't necessarily a lay summary, but it's a sort of discovery science often takes you a journey that you don't necessarily know exactly where it's going to be useful yet. The the drug stuff I was talking about right at the end, that's the direct route, right? So that is us going to say, if you have this mutation, at the end of doing that screen, this is what could be useful for you. And there are potential uh, drugs on the way that use that approach. So again, if I just share this screen, um, let me see. I've got too many screens going on here.
Yeah.
Um, so yeah, so this paper, which is a few years old now, we're using gentamicin to treat people with junctional EB, and what they did was this is the part of the skin that's been being treated, and this is a drug that basically tells the transcriptional machinery to ignore the mutation, and it looks good, and it obviously locally works well, but has a lot of side effects because the way it works isn't specific to that mutation. It'll work on lots of other things, but it tells you that there's a way, right? There's a mechanism that can be exploited to get past the problem to say, right, this isn't a mutation anymore. And so what we need is ways in which we can screen tens of thousands potentially of compounds that will allow us to then say, right, gentamicin sort of works, but isn't very specific. Let's find the one that is specific and it's going to work a bit better. And that's what I hope our next step is going to be for that. So it's sort of like the headlines is we'll know more about laminins than we'll ever know before, which means that we might identify new routes to therapy, and then we'll directly target things that we think can work.
Yeah. Do you think we'll get to a point where I'm jumping ahead of that? We could put either spray on laminin or put different parts of the lamin onto the wounds, and
Yeah.
that will hopefully help with the wound closure.
Yeah, absolutely. So some of the other people in my department, we're working together to make a bandage contact lens where we're introducing that little fragment of the laminin that I was showing in the wound healing. So that basically uh that's our next step in that part of the project, which is pretty cool. At the moment, it's really hard to make lots and lots and lots of laminin that's very pure. So getting it ready for the clinic will be a challenge. But you can buy laminin off the shelf now. So it's not ready for clinical use, but that's commercially available. So definitely possible. And there were some studies previously where people injected collagen 7 protein into the bloodstream and it got to the right areas. So the idea of a protein-based therapy has got some traction. I think that the future in a lot of these, genome editing will be the future of this. It's just getting that ready to go is kind of, it might take longer than, well, it's hard to know how long it's going to take, basically. Yeah.
No, I completely understand. It's pursuing all possible avenues.
Yeah, absolutely. I think that if genome editing it works, doing it in situ, as in straight onto a patient, might be hard still. That might be even take a long time. So the type of things that our drug screen approach would identify as something that could get used much sooner and therefore could be rolled out much sooner and hopefully could be taken locally or orally or something, which would also be much cheaper probably as well.
Yeah.
Just the other big barrier to entry.
Yeah, that I think it all comes down to quality of life. It's giving people with EB as many therapies as we possibly can to give them a normal quality of life like everybody else takes for granted. Yeah. Perfect. I don't think there's any more questions coming through.
There's a feedback form, but it'd be useful, if you could fill that in, because I get excited about talking about these things, but I never know how to pitch it, whether it was too complicated or not complicated enough, like whether you want more details or fewer.
Completely understand. And I think before people go, so first up, Kevin, thank you very much. It's been an absolute amazing webinar. I've certainly learned a lot. It's nice to see that microscopy has changed. Well, it's revolutionarily changed compared to my days when I was doing it. So I've certainly learned a lot.
Fast.
Yes, moving fast. And so before everybody leaves, I'd just like to remind everybody, please do register for our next webinar on Wednesday, the 4th of March, where we'll hear from Professor Albina Dinkino-Cosanova from University of Dundee, talk about the therapeutic potential of NERV2 activators for EB simplex. Please do register if you have not done. So, and on that note, thank you very much.